Imagine your brain, the very engine of your thoughts, your feelings, your movements. Imagine that activity could actually, unintentionally, fuel a deadly disease. It's a pretty sobering thought, isn't it? But today, we're diving into some really groundbreaking discovery that reveals this exact, unsettling reality in a devastating form of pediatric brain cancer. Our deep dive today is into diffuse midline glioma, DMG for short. It's an aggressive, often fatal cancer affecting children for just way too long. Treatments have been, well, desperately limited, but new research is shining a light, maybe, on some unexpected pathways within the brain itself.
Yeah, and for this deep dive, it's crucial we look closely at the source. We've dissected a really vital article. It's from the journal Cell, a top-tier journal, published just this August 2025. It's by Drexler and colleagues, and the title tells us a lot right away. Cholinergic neuronal activity promotes diffuse midline glioma growth through muscarinic signaling. This isn't just, another paper. It feels like a significant shift in how we might understand this terrible disease.
Absolutely. So our mission today is to really unpack the surprising role of these things called cholinergic neurons. They're a specific type of brain cell in driving DMG growth. We'll explore how these neurons influence the tumor, and maybe even more strikingly, how the tumor then seems to manipulate these very same neurons. And ultimately, we want to pinpoint specific therapeutic targets that could potentially offer some new hope.
Okay, so let's unpack this a bit. We know the nervous system, it can play a really crucial role in cancer, especially when the cancer is in the brain. Seems logical, but maybe for you, our listener, let's back up. What exactly is diffuse midline glioma, and what do we already understand about its connection to brain activity before this latest research?
Right, good place to start. So diffuse midline glioma, or DMG, it's an incredibly aggressive central nervous system cancer. It targets structures right in the midline, the very center of the brain. Think places like the pons, which is why it's often called DIPG, diffuse intrinsic pontine glioma, but also the thalamus and even the spinal cord. What makes it particularly devastating and difficult to treat is that it's largely driven by specific genetic mutations, usually in a protein called histone H3. You might see it written as H3K27M. This mutation fundamentally messes up how DNA is packaged and read inside the cell. And that directly contributes to the tumor's really aggressive nature. And it's really important to grasp this. DMG cells, they actually originate from and look a lot like healthy cells called oligodendrocyte precursor cells, OPCs for short. Think of OPCs as like the brain's immature builder cells.
Right.
Their main job is to grow up to mature into oligodendrocytes. And oligodendrocytes are the cells that produce myelin. That's the fatty insulation around nerve fibers, kind of like the plastic coating on electrical wires. It helps signals travel fast. So these cancer cells are essentially hijacking the characteristics of these vital developing brain cells, but twisting them for malignant growth. Now, before the cell paper came out, we weren't completely in the dark about brain activity in glioma. Previous research had already shown that the activity of other types of neurons, specifically glutamatergic and GABAergic neurons, could actually promote the rapid growth, the proliferation, of both healthy OPCs and these malignant glioma cells. Glutamatergic neurons, they generally use glutamate. They're kind of like the brain's on switches, exciting other cells. And GABAergic neurons use GABA. They're more like the off switches, calming activity down. So we already had this idea that the brain's electrical chatter, both the excitatory on signals and the inhibitory off signals, could somehow fuel these tumors.
Okay, so if we already knew that those other types of brain activity, the glutamate and GABA ones, were involved, why the big focus now on cholinergic neurons? What makes them different? Or why are they suddenly a critical piece of the puzzle for this disease?
That's a really sharp question, because while glutamatergic and GABAergic neurons handle, let's say, the brain's main rapid-fire communication, the role of a different class, the neuromodulatory brainstem neurons and cholinergic neurons, fall into this category. That role has largely been, unexplored in glioma growth. Neuromodulators, they don't just send simple on-off signals like the others. They sort of fine-tune vast networks of neurons. They influence broader brain states like arousal, attention, learning, memory. They're more subtle maybe, but incredibly powerful regulators. And what really makes them super relevant here is their location, their strategic position, and their connections. The main hubs, the nuclei, where these cholinergic neurons live, places like the levodorsal tegmentum nucleus, or LDT, and the pituitary-pontine nucleus, the PPN. They're located right in the midbrain. And from there, they send out these long projections, these axons, directly into those midline structures, the thalamus and the pons, precisely where DMGs most often pop up. So, they're perfectly positioned to influence whatever cells are in those regions, whether they're healthy cells or, unfortunately, tumor cells.
That positioning is key, isn't it? I like the analogy you mentioned earlier. if the brain is like this complex orchestra, then the glutamate and GABA neurons are maybe the main sections, the violins, the trumpets playing the core melody.
Right.
But these neuromodulatory neurons, like the cholinergic ones, they're more like the rhythm section, the
Mm.
drums, the percussion. They might not play the main tune, but they set the tempo, the intensity, the whole feel of the piece. And in this case, it seems like they're setting a rhythm that actually promotes cancer growth, which is, yeah, a chilling thought. Okay, quick recap for everyone listening. So diffuse midline glioma, DMG, it's this terrible pediatric brain cancer, often linked to that H3K27M mutation. Its cells come from and act like healthy OPCs, those precursor cells. We already knew other brain activity, glutamatergic and GABAergic, could fuel its growth. But today's deep dive brings a new player into focus. These cholinergic neurons, especially from the LDT and PPN in the midbrain, because they're so strategically located near the tumors. So you might be wondering, well, what exactly is a cholinergic neuron in more detail? And how on earth did the researchers even start testing if they influence cancer?
Right. So to be precise, cholinergic neurons are simply neurons whose primary chemical messenger, their neurotransmitter, is acetylcholine. For short. Acetylcholine is, well, it's famous in neuroscience. It's involved in so many things, muscle contraction at the neuromuscular junction.
Yeah.
But also in the brain, it's crucial for wakefulness, attention, learning, memory consolidation, really fundamental stuff.
Okay, and here's where the study gets really, really interesting, I think. The researchers first asked a pretty basic but crucial question. Does this cholinergic activity affect the healthy OPCs, those immature builder cells that the DMGs resemble? You kind of have to understand the normal baseline before you can figure out what's going wrong, right?
Yeah.
So what did they find with these healthy cells?
Precisely. You need that baseline. And they found a very clear, significant link. Midbrain cholinergic activity from those LDT and PPN neurons, it significantly increases the proliferation. That's the rate cells divide and multiply. So it makes healthy OPCs multiply faster. But it weren't just a general turn up the growth everywhere kind of effect. It was surprisingly precise. It was circuit dependent. They use this amazing technique called optogenetics. Briefly, it lets you control specific genetically targeted neurons just using light, like installing a light switch for specific brain cells. It's incredible. So they engineered these cholinergic neurons to be activated by blue light. When they shone blue light specifically on the LDT nucleus, remember, that one projects mostly to the thalamus, they saw an increase in OPC proliferation, mainly in the thalamus. And then when they stimulated the other main group, the PPN nucleus, which projects heavily to the pons, guess what? OPC proliferation increased specifically in the pons. It's like these cholinergic neurons are sending very targeted grow here signals to specific neighborhoods in the brain. They did see this effect in the cortex too, which received some cholinergic input, but interestingly, not in other areas they checked, like the hippocampus. So it really highlights this brain region specificity.
Wow. So healthy precursor cells definitely respond to this cholinergic activity and in a very specific, targeted circuit-by-circuit way. That's a critical finding. Now, okay, the big, maybe scarier question. What happens when you apply the same approach, the same lens, to the malignant DMG cells, these, malignant counterparts of OPCs? Do they also listen to these specific cholinergic signals?
The findings here are, well, strikingly parallel, which is both scientifically fascinating and, frankly, quite concerning from a disease perspective. It strongly reinforces this idea that the cancer cells are co-opting or hijacking a normal biological pathway for their own destructive growth. The bottom line is, DMG cells are equally responsive to this cholinergic neuronal activity as their healthy OPC counterparts are. So again, using optogenetics, they activated cholinergic nerve fibers, both the ones locally within the tumor areas and those long-range ones coming from the LDT and PPN. And consistently, across different models, they saw a robust increase in the proliferation rate, the growth speed, of DMG cells in both the pons and the thalamus. And crucially, just like with the healthy OPCs, this tumor growth boost was also circuit-dependent. PPN stimulation specifically revved up DMG growth in the pons, while LDT stimulation revved it up in the thalamus. And they didn't just show activation works. To really nail the cause and effect link, they did the opposite experiment. They used inhibitory optogenetics using a different light-sensitive protein to reduce cholinergic activity. And when they did that, they saw a circuit-specific decrease in DMG proliferation. Turning down the cholinergic signal slowed the tumor growth in that specific circuit. That's pretty powerful evidence for a direct causal role. What makes this really compelling from a translational standpoint is that these findings held up across different, quite sophisticated mouse models that used genetically engineered mouse models that spontaneously developed these gliomas, but also models where they implanted human patient-derived DMG cells directly into the mouse brain. The results were consistent. That really boosts the confidence that this might be relevant to human patients, and they could even watch the immediate effect in real time. Using something called fiber photometry, it lets you measure calcium signals, which are a good proxy for cell activity. Deep in the brain, they showed that activating cholinergic neurons increased calcium signals inside the patient-derived DMG cells. And we know that increased calcium signaling inside glioma cells is a known driver of their progression, their proliferation, and invasiveness. So this is like catching the mechanism in the act.
Okay, let's pause for a sec. Recap number 2. We've established that these specific cholinergic neurons, particularly from the LDT and PPN, acting through their precise connections, are powerfully promoting the growth, the proliferation of both healthy OPCs and these aggressive DMG tumor cells. It really looks like the glioma cells are exploiting a normal brain process, almost like they've learned the brain's secret growth language. But how? Yeah. How is this communication actually happening at the molecular level? What is the specific chemical language being spoken between these neurons and the tumors?
That's the next critical question, right? The how. The researchers dug into this and uncovered 2 main mechanisms, although one turned out to be particularly dominant in the key tumor locations, the pons and thalamus. First, they looked at paracrine factors. These are basically signaling molecules that cells release into the space around them, influencing nearby cells without direct contact, like sending a message in a bottle through the surrounding fluid. They found that activating midbrain cholinergic neurons caused them to release more of a specific factor called brain-derived neurotrophic factor, or BDNF. BDNF is a well-known growth factor in the brain. They showed this by stimulating cholinergic neurons in brain slices kept alive in a dish, these are called explants, and then collecting the surrounding fluid, the conditioned media. This BDNF-rich media, when they put it onto DMG cells grown in culture, made the DMG cells proliferate more. And even more promising for potential treatments, when they used a drug that blocks the receptor for BDNF, that receptor is called TRAB, they saw a decrease in glioma cell proliferation in vivo, in living mice. This suggests the BDNF-TOE-AKB pathway is one targetable route. However, and this is a key finding, while BDNF seems to play a role, especially maybe near where the cholinergic cell bodies are in the midbrain, further experiments revealed an even more direct and probably more dominant player, especially right where the tumors grow most often in the pons and thalamus. And this brings us to the second, and likely most critical, mechanism. Direct acetylcholine, ACA, signaling via muscarinic receptors. This means the acetylcholine neurotransmitter itself, released from the cholinergic nerve endings, can directly act on the DMG cells. They demonstrated this quite elegantly. They grew human cholinergic neurons, derived from stem cells, together with patient-derived DMG cells in a dish. Just having them together increased DMG proliferation. And when they used optogenetics to specifically activate only the cholinergic neurons in that co-culture, the DMG proliferation increased even more. They even found electron microscopy evidence suggesting structures that look like synapses, those specialized junctions for communication forming between the cholinergic neurons and the DMG cells. That implies really direct, maybe even fast, communication. To really confirm this direct action of AKs, they just took DMG cells in culture and directly added acetylcholine to them at different concentrations. And they saw a clear dose-dependent increase in proliferation. More acetylcholine meant more tumor cell division. That's a very strong indicator of direct causal action by AK itself. Okay, so AK is the signal. What's the receiver on the tumor cell? The receptor. They found that the growth-promoting effect of acetylcholine was specifically blocked by drugs that inhibit muscarinic receptors. But it wasn't blocked by drugs that inhibit nicotinic receptors, the other main type of AK receptor. Furthermore, an agonist drug that mimics AK and muscarinic receptors called muscarine also increased proliferation, while nicotinic agonist, like nicotine itself, did not. This really pinpoints muscarinic receptors as the culprits on the tumor cells. Then came the molecular confirmation. They looked at gene expression data using single-cell RNA sequencing from actual primary human DMG patient samples. And bingo! They found high expression of the genes for 2 specific muscarinic receptors, CHRM1 and CHRM3. These genes code for the M1 and M3 muscarinic receptors. And crucially, this high expression was seen particularly in those OPC-like malignant cells within the DMG tumors. It's the molecular smoking gun, showing these targets are right there on the cancer cells. And here's a critical detail for potential therapies. They found these M1 and M3 receptors seem to play a somewhat redundant but absolutely vital role. Blocking just M1 or just M3 wasn't enough to completely stop the cholinergic-driven proliferation. They had to block both M1 and M3 together, either using a combination of drugs or by using CRISPR gene editing to knock out both genes in the DMT cells. Only blocking both could fully halt the effect, both in culture and in mice. This redundancy really emphasizes how critical these specific receptors are for the tumor's response to acetylcholine. And it wasn't just about proliferation, about making more tumor cells. They also found that exposing DMG cells to acetylcholine significantly increased their migration in lab tests. That suggests MD might also contribute to the tumor's ability to spread or invade surrounding brain tissue, which is obviously a key feature of aggressive cancers. And one final important point on mechanism. They did experiments to show that these cholinergic effects were happening independently of the glutamate or GABA signaling pathways we mentioned earlier. So this is definitely a distinct, separate pathway promoting glioma growth that hadn't been fully recognized before.
Okay, wow. So let's get this straight. We're seeing not just, general growth factors like BDNF playing a role, but direct signaling. Acetylcholine itself, released by these specific neurons, acts directly on these M1 and M3 muscarinic receptors located right on the DMG cells. And this drives not only tumor proliferation, more cancer cells, but also their migration, their ability to move. And it's a distinct pathway, separate from the ones we already knew about. That's a massive discovery. But is this just a one-way street? The neurons signal, the tumor grows, or does the tumor somehow talk back? Does it influence the brain, creating, I don't know, some kind of truly vicious cycle?
This is where the story takes another really powerful and frankly quite unsettling turn. They discovered exactly that, a bidirectional interaction, a vicious cycle. It turns out DMGs don't just passively respond to cholinergic activity, they actively increase it. They essentially pour gasoline on their own fire. The evidence for this tumor talks-back phenomenon is pretty strong, coming from multiple angles. First, they looked at COFO's expression in the brains of mice with tumors versus healthy mice. Remember, CHOS is a marker protein that indicates recent neuronal activity. They found significantly increased CFO's expression specifically in those LDT and PPN cholinergic nuclei in the tumor-bearing mice. This means those neurons were more active simply because the tumor was present elsewhere in the brain. Then they went further and did direct electrophysiological recordings from individual LDT cholinergic neurons in mice with tumors. These recordings showed that the neurons in tumor-bearing mice were intrinsically more excitable. They fired more readily, had more spontaneous activity, and stronger responses to stimulation compared to neurons in healthy mice. The tumor was clearly changing how these neurons behaved. And they used those sophisticated sensors again for real-time monitoring in the tumor environment. Using a sensor designed to detect acetylcholine itself, it's called GRAB HE 3.0. They measured HE levels right in the tumor microenvironment over several weeks as the tumor grew. They saw a significant and progressively increasing release of acetylcholine over the course of the disease. As the tumor got bigger, it triggered more and more HE release right around it. Finally, using fiber photometry again, but this time measuring calcium signals back in the LDT and PPN neuron cell bodies, not the tumor cells, they saw elevated calcium transients there too. This indicated increased activity in the cholinergic neurons themselves, and this activity also increased as the tumor progressed. It provided a real-time view of these neurons becoming hyperactive as the glioma grew larger.
That's a really critical insight. It completely changes the picture. It's not just a one-way command from neuron to tumor. It's a feedback loop. The tumor isn't just this silent, passive mass responding to brain signals. It's actively manipulating the brain environment around it, amplifying the very signals that are helping it grow. It's a truly insidious, self-reinforcing mechanism. It's almost like a parasitic relationship, but way more complex and dangerous, embedded right within the brain's own circuitry.
Exactly. And if we try to connect this back to the bigger picture for the patients, this bidirectional interaction, this vicious cycle, it has potentially profound implications. We have cholinergic activity promoting tumor growth, and then the tumor in turn amplifying that cholinergic activity, just keeps feeding itself. This raises another important question. What are the broader consequences of this tumor-induced hyperactivity, this dysregulation of these crucial cholinergic neurons for the patient's overall well-being, beyond just making the tumor grow faster? We know that DMG patients, tragically, often experience significant non-motor symptoms. Things like cognitive difficulties, changes in behavior, emotional disturbances. It adds terribly to the burden of the disease. And these brainstem cholinergic neurons, particularly in the LDT and PPN, they're known to play really important roles in regulating things like attention, arousal, sleep-wake cycles, and even aspects of cognition and mood through their widespread neuromodulatory influence. So, this study strongly suggests or proposes that the tumor-induced dysregulation and hyperactivity of these specific brainstem cholinergic neurons could be directly contributing to some of these debilitating neurological and cognitive symptoms seen in patients. It highlights why understanding these neuron-tumor interactions is vital not just for finding ways to stop tumor growth, but potentially also for improving the patient's quality of life during the disease.
Wow, okay, what a deep dive that was. We've really uncovered a truly fascinating and yet alarming interaction between these midbrain cholinergic neurons and diffuse midline gliomas. We've seen how specific brain circuits using acetylcholine acting on these M1 and M3 muscarinic receptors can drive tumor growth and even spread. And then the really stunning part, how the tumor seems to fight back, amplifying that very neuronal activity that fuels it. This feels like more than just groundbreaking science. It feels like a genuinely vital step in understanding a devastating disease that has just seemed so intractable for far too long.
I agree. The study by Drexler and colleagues, it really does fundamentally shift our understanding of DMG pathophysiology. It adds another critical layer. It shows that, maybe each major neuron type studied so far, glutamatergic, bidirectional, now cholinergic, contributes to DMG progression, but through distinct mechanisms. And importantly, mechanisms that seem targetable. The evidence pointing to M1 and M3 muscarinic receptors as potential therapeutic targets is incredibly promising. And as you hinted, this might extend beyond just DMG. Cholinergic signaling, often involving these same muscarinic receptors, has been implicated in other cancer types too, gastric, colon, prostate cancers, among others. So the implications could be quite broad. It really opens up a whole new avenue for investigation and hopefully treatment development.
Absolutely. It moves us closer to a more complete picture of what some people are calling the neuroscience of cancer. Understanding how the nervous system, both within the brain and maybe even peripherally, interacts with tumors. And hopefully, towards more effective combination therapies for children and young adults facing these lethal CNS cancers, maybe targeting multiple pathways at once. It also just reminds us, doesn't it, that the brain's incredible complexity is often right at the heart of both our greatest health challenges, but also potentially our greatest breakthroughs. So for you, our curious listener, maybe here's something to ponder as we wrap up. If cancer, like DMG, can so cleverly hijack and amplify specific neural circuits in the brain for its own growth, what other subtle, maybe currently invisible neuronal conversations might be happening between nerves and tumors in different types of cancer, maybe even elsewhere in the body? And how might understanding those intricate conversations lead to entirely new, perhaps more precise ways to diagnose, monitor, and ultimately fight disease? What really stands out to you from today's deep dive, and what new questions does it spark in your mind?